All right, so let's review meiosis. Everything starts in interphase. Okay, this is before any cell division. Um, know that during interphase we have this chromosomes duplication, so we have DNA replication. This happens in the S phase. Okay, and at this time, let's think about it probably a little bit. Probably in the late G2 phase, the chromosomes are still uncondensed, so you really cannot tell them apart. All right, and then we start with meiosis one. This is meiosis one. We start with one cell. This cell is going to be diploid. Okay, here's our cell. One cell diploid, and this imaginary cell has four chromosomes. Okay, four chromosomes. Prophase one, what happens is, so all of these chromosomes have sister chromatids. Okay, and they're going to pair up. So pair homologous chromosomes are going to pair up. They pair up in something. This is the pairing up, and the pair up is usually referred to as a tetrad because it has four sister chromatids. So that's a tetrad. Um, Homologous chromosomes pair up, and they exchange segments. And this exchange of segments, okay, I'm going to go over here, sites of cross. It's going to be called crossing over. And crossing over is very, very important because this is one of the reasons how meiosis is going to add some genetic variation to your chromosomes. Okay, this is how we gain genetic variation. So all of this happens in prophase one. All right, we have something called crossing over because the pair of homologs are together, they get together, and some chromosomes, okay, are going to do something called crossing over. We'll talk about that in more detail in another slide. Then we go to metaphase one, and in metaphase one, like I've been saying, you think about metaphase for middle. In metaphase one, you have the spindle tracks or spindle fibers attaching to the chromosomes, but they're attaching to homologous chromosomes. Homologous chromosomes. Okay, they attach into the homologous chromosomes. You can see it here how this spindle fiber is actually attaching to this one, right? And this one is attaching to this one. Okay, and also on the other side, we have this spindle fiber attaching to this one, and this one attaching to this one. Okay, so spindle tracks attached to the chromosomes. Uh, the chromosomes are still pairs of chromosomes are starting to line up in the middle. Okay, this is the middle of the cell plate or the middle of the cell. Then we go through anaphase one, and like I said before, A for away. So something's gonna happen that chromosomes are gonna move away. In this case, the homologous chromosomes, and this is kind of the important thing, right? The pairs of homologous chromosomes split up. All right. So this homologous chromosome is gonna go into this direction. This one goes into this direction. This one goes in that direction. That one goes into that direction. They're going to opposite poles. Okay. Um, going to opposite poles. The important about this is this, guys. Okay, and please keep this straight. Uh, Homologous chromosomes separate. Homologous chromosomes still with sister chromatids. Okay, they're still together. The sister chromatids are together with sister chromatids separate. Okay, so homologous chromosomes separate, and then we end up with in telophase one after meiosis one or in meiosis one, telophase one and cytokinesis one. Okay, and we end up in this phase. Now we have two cells. All right, and each of them are going to be haploid. Okay, why? Because the homologous chromosomes separate. So now this cell here, this one is going to be haploid because it has two chromosomes. Okay, and this one also has two chromosomes. Okay, so what we see here is in meiosis one, we go from diploid to haploid. And also, in meiosis 1, we go through some genetic variation, which you will see as crossing over, and we'll see as independent assortment in metaphase 1. We'll talk about that later. And then we go to meiosis 2, and in meiosis 2, we start again, we start with whatever we ended up, right? So I'm sorry, I want to go back to my previous slide. We end here. It wasn't really an end. Okay, I don't know if you ever gone to a theater production, but in the theater production, um, in a play, you usually have kind of act one, right? And then you have act two, right? So think about that. Act one being meiosis one, and Act Two being meiosis two. Okay, so whatever we're left off in Act One, that's what we start in Act Two. So we have two chromosomes um, in each cell, two haploid cells. Chromosomes are still doubled. What does that mean? Chromosomes still have sister chromatids. Okay, so from here, now we go to meiosis 2. In meiosis 2, um, kind of the big important thing that happens. Um, kind of the big important thing that happens is, well, before we start here, just remember this: no DNA replication happens here. No DNA replication before meiosis 2. Getting a little sloppy here. Before no DNA replication before meiosis 2. Okay, we start with two cells, right? Just where we ended, we start with two cells. Both cells are going to be haploid. Both cells have two chromosomes each. Okay. Now both chromosomes have sister chromatids. All right. Now what happens in metaphase 2? Again, M4 middle. So something happens to the chromosomes that they're going to gather in the middle. Okay. And look at that way they are in the middle. Now they are lined up, just like in mitosis. Now, this is not mitosis, okay? This is just to show you how they line up exactly like in mitosis. And then this is your spindle fibers. Don't forget that, your spindle fibers. Okay, spindle fibers. Now, in anaphase, what happens in anaphase? A4 away, right? So something happens to a chromosome, they split. Now, this is the important thing about meiosis too. Sister chromatids separate. Okay, this sister chromatids go this direction, this one goes in this direction, this one goes into this direction, this one goes to that direction. But the key word is this. Okay, in my anaphase 2, I'm going to repeat it. It's so important, I'm going to repeat it again. Sister chromatids separate. Okay, there you go. That's how important it is. At the end of the day, after telophase 2 and cytokinesis, what happens? Well, we end up with 4 cells. Each cell is going to be haploid. Okay, but now we have 2 cells. They're haploid. Each chromosome, each cell has 2 chromosomes. Okay, we started with 1 cell, 4 chromosomes. We end up with 4 cells and 2 chromosomes, and that is meiosis.